I will start. Yeah, thanks a lot for the invitation. Yeah, sorry everyone for the initial, I think the sound issues with the host. But yeah, I'm glad people can hear me. And my topic today is about development of ultrasound-activated nanoparticles for non-invasive genetic targeted neuromodulation. And I'm Evan, assistant professor at UT Austin Biomedical Engineering Department. So I hope to give you first some motivation for this work. So as many of you know, deep brain stimulation has been used for treating neurological disease, especially Parkinson's disease, but also extending to epilepsy, psychiatric disease. However, the downside of this technology, right, is stimulating all the neurons in the brain area, so it's not targeted, so not desirable for some of the more complicated disease. And can potentially cause side effects. In comparison, there's also technology called optogenetics, where we can express light-sensitive opsons in specific type of neurons. So in this case, in comparison to the electrical stimulation, like a deep brain stimulation that stimulates all the neurons in the brain area, optogenetics, we can selectively stimulate specific type of neurons in the brain area, since we can express these opsons in only certain type of neurons. And so as a result, we can either use blue light to excite or yellow light to inhibit. And there are also many other options that can be used for selectively modulating specific type of neurons. So here are some examples where we can use blue light to activate neurons at a specific frequency, like 5 hertz, 30 hertz. And in this case, where we have a mice with implantation of a fiber, where we can apply blue light for 20 hertz, and then to get the mice to rotate in a specific direction, since we only implant the fiber into one side of the brain. And here is another example where we express a halo rhodopsin, like yellow light activated opsons into the neurons, and you can see that we can inhibit the firing of the neurons with yellow light. And here is a case where we have a worm swimming around, and then we can use yellow light to inhibit the movement of the worms and that's an example of inhibitions. So in comparison to the electrical stimulation, optogenetics does so advantage in comparison for like disease treatments. In this example, if we stimulate all the neurons in this GPE brain area, it does not like give like a more like a longer term effect for the Parkinson disease. As we know, like for deep brain stimulation, for example, we only give like symptoms relief, not like over time like effects. So here as well, like if you see if we shine the blue light, you can see we can get the recovery of the reducing the immobilities of the animals only when we hit with the blue light. But after stop when we stop hitting with blue light, the immobility will increase again. But in this case, if we only stimulate PV neurons in this brain area, you can see the effect is cumulative. So basically, like even if we stop doing stimulation, there's also effects like lasting for a long time. So that's really showing the advantage of the cell-specific stimulation for longer therapeutic effects. And here is another example. I don't showing the result, but yeah, just like to really showing like in this paper, if they are only stimulating a specific projection, it can also recover the effect for the depression model. But then if they stimulate all the neurons in the brain areas or not projection specific, they don't get see this type of effects. So, however, there's also downside with optogenetics is that it requires implantation of a fiber into the brain. In a similar case with deep brain stimulation as well. So basically, especially if we want to stimulate places deep in the brain, you can see we need to implant the fibers, so like to drill a hole in the brain and implant the fibers all the way in the deep brain areas, and all the neurons along the fiber track will be destroyed. So it could be damaging to the brain tissues and the blood vessels sometimes and all that. So basically, we probably know that the ultrasound has been used for look at babies, right, like to use widely in the clinic. It has a really good penetration of up to 20 centimeters, so it's really good for non-invasive clinical applications. And here we also know that there are nanoparticles, piezoelectric type of nanoparticles. That when we apply mechanical force, it can emit light. So this like with specific type of particle compositions. So we ultrasound is kind of a mechanical wave, so you can think about it as a mechanical force. So we are basically the technology we are. When I was a postdoc at Stanford, me and work with Guozhong's lab, we have thought about to use this type of nanoparticles to be also be activated by ultrasound for light emission to do optogenetic stimulations to really enable non-invasive optogenetic neuromodulations. So basically what we do is that we inject this type of nanoparticles through the tail vein injections. We first need to charge the nanoparticles with more like a short wavelength of light when the nanoparticles gets to the neck, and then once it gets to the brain, we apply the ultrasound, and then to get nanoparticle emit blue light for optogenetic neuromodulations. So here are the case where you can see when we inject nanoparticles through the tail vein injection and apply the ultrasound at the brain, you can see the synchronized leg movement with the ultrasound, it only happens when we have all the opsons, nanoparticles, and ultrasound cases. So not in other cases, we can see pretty synchronized leg movements. So that's really demonstrating the success of this technology for sono-optogenetic neuromodulations. However, with those type of nanoparticles I mentioned earlier, it requires charging of nanoparticles, so it's not very convenient in many cases. We need to recharge and recharge, and also they are inorganic nanoparticles, so they are generally have a potentially like effects like in the body to stay in the body for a long period of time. So that's also not desirable. So since I started at UT as a assistant professor now, like almost 5 years ago, we have been developing organic nanoparticles for similar purpose. Especially in this case, we are applying liposome. So we know that has been used for delivery of mRNA for COVID vaccine, right? So we know this type of nanoparticle are very health friendly and not causing too much of the toxicity effects. So in this case, we also use similar type of nanoparticle, but instead encapsulation of mRNA, we encapsulate two type of organic molecules. One type of organic molecule is a sonar sensitizer, so that can be activated by ultrasound for generation of reactive oxygen species, RS. And the other type of molecule is a chemoluminescent molecule that can be activated by RS for light emission. So basically, with these systems, we can basically apply the ultrasound and then to get light emission right away. We don't need to charge with of nanoparticles with another wavelength of light beforehand, and we can get repetitive light emissions, and then it's much more friendly in terms of the toxicities. So here we are showing this type of nanoparticles. We can see the size of nanoparticles is around 100 nanometers in diameters. And here are showing the TM images of these nanoparticles. And here we are showing like when we apply the ultrasound, we can get the light emission with this type of nanoparticles over here synchronized when we apply with ultrasound. So here again, we are doing a similar experiments that we did before. We inject nanoparticles through tail vein injections, and when it gets to the brain, we apply ultrasound to activate those nanoparticles in the motor cortex, and then we use deep lab cut to look at the movement of the leg in these animals as we apply ultrasound. And again, over here you can see when we apply the ultrasound at a certain frequency, like 1 Hertz in this case, we can see the synchronized movement of the leg of the mice over here. And if we don't have either ultrasound, nanoparticles, or opsons, we don't see this type of movement. So it's again really demonstrating this technology can work for neuromodulations. So that's just to in order to confirm more, we get neuroactivation. We also take the brain tissues, we slice them, and then we can again to see the C-force expression. So that's basically if the neurons get activated, we see this. We can stain the C-force and to see the C-force expression in this brain regions, just to confirm the neural activation, and we can see again like we need all the components to see the C-force expression. If you don't have one of those, we don't see the C-force expression. And the next step, so we have demonstrated in sono-optogenetic neuromodulation in the motor cortex, like more superficial, like brain regions. Now we are hoping to go deeper, right, to demonstrating the deep brain neuromodulations. And in order to do that, we would like to the nanoparticle to emit like a even higher intensity of the light. So then basically it's more effective to activating of the deep brain areas. So in this case, we designed the we still have our liposome nanoparticles, the same sonosensitizer, chemoluminescent molecules, but we also added one component of the calcium oxide. In this case, when we hit with the ultrasound, the calcium oxide can do two things. One thing is to generate more ROS, reactive oxygen species, and then it also generates calcium hydroxide. So the function of that is basically to increase the pH of the system, because if we have too low of the pH, it will quench the chemoluminescent molecules. So if we increase the pH, it can prevent the quenching, so makes the light emission more efficient with these chemoluminescent molecules. So as you can see over here, we are able to get like higher light intensity with this new type of nanoparticles with same concentration and same ultrasound pressures, and also we can really demonstrate we can really apply the ultrasound at different frequency to get the light emission at different frequencies, and also the latency between the light emission from the application of the ultrasound is quite small. It's only a few seconds. Sorry, only a few milliseconds. So really showing like very fast speed light emissions. And here, in this case, we are demonstrating this technology can activate deep brain areas in the mice. In this case, we have the we are looking at the area called the BTA. That's a area related to addictions, and in this case we inject the opsins and nanoparticles in these brain areas, and basically like when we activate this area, the mice will be happy, will like this area more. So the behavior task we are using is called the lever press behavior. So basically when the mice press the lever, it gets ultrasound, and then basically if it is get success in stimulation, the mice will like this and then to press more. So here is of the videos to showing that basically the mice doing the lever press behaviors, and then the three of them are controls, and only the bottom right are the mice which like our active groups. So you can see basically over here as the mice is like pressing the levers right more, and then it gets a stimulation. So basically the mice will like it more and then press it more. So yeah, that's really showing like the behavior response. And so here's what it looks like, and here showing the number of times the mice press the lever. So that's before. There's no neuromodulation, yeah, no ultrasound activation. During is when the mice press, we get the activation. So then you can see our experimental groups, the mice will press much more in comparison to other groups which without one of the components. And so we did it over 3 days, and then afterwards, so we remove the ultrasound activation again. There's still a residual effects, right? Because the mice already knows that when they get press the lever and it gets a stimulation. So there's still like it still press more than the other groups. Yeah, that's really to demonstrating we can activate the VTA of the mice with this our sono-optogenetic approach, and the next in order to basically we are also moving like from the smaller animals to larger, like going from the mice to rats, and also we are also plan to do it in the more diseased case to demonstrate that our technology can be used for disease treatment. So in this case, since we're moving to rats, we want the nanoparticles to be able to be even more efficient at emitting light and also emitting light for longer period of the time. So in this case, we are using a different type of nanoparticle called the hydrogen bonded organic framework. So those are like a three-dimensional materials that has a three-dimensional pores in there, so it basically can load. Like you can see, the loading capacity is almost like 4 times, or sorry, more than 4 times in comparison to the liposome case. It can load much more molecules, and in addition, this organic framework itself can also act as a sonosensitizer. They can be activated by ultrasound to generation of our acid itself. So then we don't need to load any more of the other sound sensitizer. We can efficiently use all the ports to load the chemoluminescent molecules. And again, over here, we can see like we can get efficient light emission when we apply ultrasound, and the light intensity is much higher than the liposome case, and we can do it the repetitive light emission for a much longer period of time with this approach. And here, so here we are showing like the size of the mice brain and the rat brain. The rat brain is much larger, right? So in this case, we are doing it in the rat, and we are looking at the area I showed like early in my introductions to, that's a brain area related to Parkinson disease. We are, if we stimulate the PV neuron in the GPE region, right, we are showing like the longer term therapeutic effect, not just the symptoms release. So basically over here, we are trying to verify we can activate neurons in the GPE region. So basically over here, we first inject the virus and nanoparticles in this areas. And then we use GI gecko to record the calcium activities with fiber photometry for the recording. So we use the CHR2 to stimulate the neurons, and we can see as soon as we apply the ultrasound, we can see the increase in the neural activities through this calcium imaging with the fiber photometries from RWD, and then we are indeed like only showing this increase when we have all these components, but not in other components, and we are able to see the pretty clear increase in the neural activation over here. And so having demonstrated that we can activate neurons from the GPE region, now we are basically doing it in a transgenic, like basically rats of the PV Cree rats, and then we express these options in the PV neurons in those rats, and yeah, in this case, you can see that those are the PV neurons. These are options. We can see we can activate the PV neurons in these brain areas, and if we look at the CFOS expression, so there's activation of the neurons over here, and then we are doing the behavior effect to see if we do get the therapeutic effect for the Parkinson applications. So in this case, where we have the rats, we inject the C6-OHDA, so that is a drug that will damage the dopamine neurons in the brain. So basically, it's like a way to create Parkinson model. So the Parkinsonian model is like basically we damage the dopamine neurons from one side of the brain. So basically, in this case, when we put the rat in the cylinder, right? So if we damage one side of the dopamine neurons, it will only use one hand to touch the cylinder walls. They will only use so basically, here is like the behavior that's counting the percentage of counterlateral touches on the walls. So basically, with the healthy rats, it's 50%. They are using both paws to touching, right? But then with the Parkinsonian, Parkinsonian rats, it only just use one paw, the healthy ones. So then it becomes around 0%. So in this case, when we do the sono-optogenetic stimulation of the PV neurons at the GPE areas, you can see we are recovering the percentage of the contralateral touch in the rats, right? more towards a healthy case, and it does last for some time, like at least for 15 minutes, right? So it does has some therapeutic effects, and especially if we compare that with just doing optogenetic stimulation without nanoparticle ultrasound with implantation of fiber, the effects are very similar with our sono-optogenetic approach, right? So it's really showing our efficacy of our technologies similar to optogenetic neuromodulation for treating of the Parkinson disease. And here's another behavior we are running is that with this Parkinsonian Parkinsonian model, the rat will keep rotates, right, when we put into the cylinders as well, when we inject the apoptosis. But then if we do the neuromodulation, right, so we can see it really reduce the amount of the rotations. So you can see with the Parkinsonian rats, the rotation speed is very high. With healthy rats, it's very low. Rats don't normally rotate, and then if we do the our sono-optogenetic stimulation, really reducing the angular speed, and the effect is also very similar to what we do with optogenetic stimulations. So it has a similar therapeutic effects as optogenetics. So yeah, basically that's a demonstration of our technology for sono-optogenetic neuromodulation in the diseased rats, and the next so we all know that the optogenetics we use opsins, right? So the opsins are generally actually coming from the like algae or bacteria or those places. So one issues of that is that it can potentially cause immune response, especially when we translate the application in humans, right? Because it's coming from a very different species. And there's also, if we talk about doing genetically targeted neuromodulation, there's also another way we can do the targeted neuromodulation, which is called chemogenetics. In this case, rather than use opsins, we use dreads, like a HM3D type of dreads that can be activated instead of light, but activated by drugs like CNO that can also be used for genetic targeted neuromodulations. So in this case, the dreads, they are generally coming from the like a very similar proteins of the human GPCR proteins, probably one or two mutations from human GPCR proteins. So basically, they are very similar to human proteins, and then they potentially have a less immune response in comparison to opsins. However, the downside of this technology is that it takes quite a long time for the neuroactivation if we just inject the signals through systematic injections. So it's not ideal for many of the neuro applications. So basically, in this case, we saw then since we can use our Hopf nanoparticles activated by ultrasound for light emission, we are thinking about like, can we just make the strength of the Hopf nanoparticles to be weaker? So then we can also use ultrasound to trigger the release of the chemicals like CNOs, also from these Hopf nanoparticles, and then to activating of the dreads. So that'd be a potentially more biocompatible method for the neuromodulations. So basically, here indeed we are going from like this previous half which like more hydrogen bonding, more pi interactions due to the fused rings. We basically make it less hydrogen bondings and also like less fused rings to make it be easier to be activated by ultrasound for drug release. And here indeed in this case, the ones we used for sono-optogenetic stimulation, it requires much higher ultrasound pressure for release of the molecules. But with this TATB half, we can use much lower ultrasound energy, ultrasound pressure for release of those drugs. You can see the threshold pressure is much lower for the release of the molecules. And indeed, from all these different type of half, we can also really compare them, and we indeed found that if there's more hydrogen bonding, more pi interactions, we get higher cohesive energy and higher ultrasound threshold for drug release. So we can actually build a map to really get a more programmable, like customized, like design according to the need of the ultrasound threshold energies. So basically having demonstrating that, we now think about like, yeah, let's apply it, right? Let's apply the most sensitive half nanoparticles for the neuromodulation. So we first to look at if we are able to get the release and also like a repetitive release with these technologies. And here are nanoparticles, and here we basically we can see only when we have the ultrasound, we can get the efficient release. And if we apply for different ultrasound pressure, we can get different amount of the release, and we can get pretty good repetitive release with this ultrasound activation and the drug release. And then we are doing for the neural activation. We first demonstrated in the cultured neuron systems. We cultured hippocampal, we cultured primary neurons over here, and then we basically have the our half nanoparticle encapsulated with the CNOs on there, and we apply the ultrasound to cause the release of the nanoparticles. And here we are using the calcium imaging to measure the firing of the action potentials from these cultured neurons. And you can see over here, when we apply the ultrasound, we can see the neuron would start to fire when we apply the ultrasound. And this is only happens when we have these all the components of nanoparticles, opsons, and ultrasound. If we don't have one of the component, we don't get this type of firings. And here we are also demonstrating it's actually pretty fast, only like around 2 seconds when we apply the ultrasound to the release of the dress. So it's much faster than the conventional chemogenetics. And in this case, we next to move to the in vivo applications to animals. We again to do it in the mice VTA. Over here, we again use fiber photometry to demonstrate we are able to get the very good neuron activations when we apply the ultrasound to release the CNOs to activation dress. And then it only takes around like also like a three or 4 seconds. So it's also very fast in vivo applications for the release. And here we again to confirm with the C-force expression, we want to get a C-force expression. We have all the components. Here's an analysis over here to demonstrate the case. So this is really demonstrating like at the VTA, we can really activate nanoparticles deep in the mice brain to for the release of the CNOs for chemogenetic applications. And here we also did like a place preference behavior in the mice. So that's another behavior related to the activation of the VTA. Basically, we know that the VTA can induce the reward and make the mice like certain things. So in this case, we let mice to run between two chambers, and during the training phases, when we put the mice into one of the chambers, and we apply ultrasound to activate the drug release at VTA. So basically the mice will like these chambers much more. And then if we run this place preference again, the mice will stay longer in this chamber in comparison to the first chamber, and which we indeed get that in our results. You can see over here, like basically the mice will switch the preference of the mice from the previous case and to the later case to more into these chambers. And then here's the data to showing that there's an increase in mice staying in the stimulation chambers afterwards. So that's and the preference score is really just like to showing the amount of the time mice stayed in the preferred chambers, stimulation chambers in comparison to the other chambers. So there's an increase when we have all the components. If we don't have one of the components, we don't see this change. And another behavior experiment we do, again, still stimulation at the VTA, is called forced swim behavior. So in this case, we let mice to swim in the cylinders, and then basically for the cases where if we don't activate the VTA neurons, so in those cases, the mice will slow down, and then they are not really motivated, rather they're more in a depressive state. It's only in this case where in the experimental group, when we have the release of the CNO to activate the neurons at the VTA, the mice will keep swimming. They will be more motivated. They are so it's like a kind of like depression type of models, and we are able to see these changes like basically like the only when we have all the components, you can see the mice would keep swimming, keep motivated. But in the other cases where the mice would be more like giving up, like a more passive state over here. Here are just showing some of the data to really showing that the bottom one is lowest immobility volumes, and then the same over here. The other ones are more more in an immobile state. And the last thing we did with this technology is to apply them in rats as well. So again, over here, we indeed also observed the neural activation at the VTA with our sono-optogenetic technology. So this is 9 millimeters from the top, so it's very deep in the brain, and we can again to see the neural activations. We see the good CFOS expressions to really demonstrate in this activation deep in the rats, and here we are again to show in the place preference behavior as well, to show like we can also modulate the place preference behavior by activating the VTA in the rats. We can see the increase in the time the rat stayed in the stimulation chamber and the increase in the preference score. So this is really to demonstrating we can modulate like a deep in the rats at the VTA, and to also to observe both the neural activations through fiber photometry as well as the behavior with the place preference behavior. Just to summarize, so we first have the invasive optogenetics, then we at Stanford with Guozong's lab, we start to use the inorganic nanoparticles with ultrasound for modulation of the neural activity with sono-optogenetics, and then in my own lab we are basically making the nanoparticles to be more biocompatible. It does not require charging with the new type of liposome-based nanoparticles, and we increase the light emission to achieve like deep brain sono-optogenetic stimulations, as well as to use it for rats, right? Stimulate deep brain in rats, and also to demonstrate the treatment of the disease of the Parkinson disease over here with Hoff nanoparticles. And finally, we moved from sono-optogenetics to sono-chemogenetics by using the a dress, so which potentially have a less immune response. I mean, the downside of that is it's still slower than the sono-optogenetics, but potentially have a better effect for the immune response. But we are demonstrating we can activate in the 9 millimeters from the top of the rats to demonstrating the deep brain stimulation of the behavior control over here. And I think that's all I would like to talk about. So Huiliang is certainly my postdoc in our lab leading all this work together with Kevin, Ilya, and others. We're collaborating with group from Guozong from Stanford, Bang Chen from UT San Antonio, and also thanks to all the funding source and my lab. And yeah, thanks a lot again for invitation from RWD to speak over here. Great.
Okay, thanks to Professor Wang, wonderful sharing. Please take a break, and we'll invite you back to ask the questions later in the Q&A section. We'll set aside a few minutes for you to write down your questions in the Q&A section. Neuroscience research needs the help of good experimental instruments in addition to excellent tools like the spanning functional nanomaterial developed by Professor Wang. Notably, our fiber photometry device plays a key role in validating this innovation, providing a real-time detection of neural activity in awake, freely moving animals, directly confirming the efficacy of these ultrasound-activated nanomaterials. Now let's have a brief introduction to RWD Life Science. RWD provides neural signal research solutions, including fiber photometry system, optogenetic system, stereotaxy instrument, a nesting cell machine, nanoliter injection pump, and micropatapula are in hot trials now. Currently, the RWD's fiber photometry system can be used in scenarios such as calcium ion and neurotransmitter signal recordings, neurocircuit research, neurological diseases research, and crop development detection. In recent years, The use of fiber photometry to detect neural activity in the neural systems of awake, free-moving animals has become an important technique for studying emotion and behavior, neuroplasticity, neurodegenerative diseases, and validating the effects of novel neuromodulative tools. We have multi-channel fiber photometry combined with optogenetics and four-color multi-channel fiber photometry system, and has the following advantage. One, quick grips, professional data analysis, high integration, and strong expansion. RWD's fiber photometry has worked in over 500 labs, and we have users from all over the world, such as MIT, Stanford University, Duke University, the University of Texas at Austin, and so on. Welcome everyone to search our website and have a try. Now it's time for the Q&A section. You're welcome to ask questions in the Q&A box. Professor Wang will choose the questions to answer for about 15 minutes. And the questioners will also have a chance to get the fiber optic cannula as a gift. Now let's give the time completely to Professor Wang.
Great. Yeah, I can see two questions from the chat. So yeah, we will certainly happy to answer those. And yeah, we'll read out the questions first before I answer the questions. So I think the first one is besides rodents, has this tool been tested on other species? For instance, has it been tested on non-human primates or other species? And have there been any considerations to validate it in vivo non-human primate models? Yeah, indeed, that's a very good question. So, yeah, we are, yes, that's certainly what we are working right now. At the moment, we are currently collaborating with yeah, group that does the non-human primates. So as you can imagine, that those experiments take long time, and they, from the like iCook Pro to do operating of the and doing the experiments, and so it does take a while. But yeah, we are certainly like working with the groups and trying to apply this technology for non-human primates. That's certainly our next step, and so certainly very good questions. Um, and I guess the next question, are the result of ultrasound pressure and drug release consistent across different brain tissues? Is there any deviation in regular precision caused by individual differences, uh, brain region structure, tissue elasticity? Is it necessary to customize exclusive half nanoparticles for specific regions? So yeah, that's also a very good question, very good thought. That's something I actually haven't thought a lot, but from my understanding is that the release of these nanoparticles will really depending on the nanoparticle structures rather than the surrounding environments. So basically, if we have the yeah for the different nanoparticles I showed, so those are the ones that really controls the threshold of the drug release when we apply ultrasound. So those TATB, for example, is most sensitive, requires less ultrasound energy pressure to release, but then the other ones will have a higher ultrasound threshold. So I would not think that different brain region would have effects, because I know the different brain region probably have some a little bit different mechanical properties, right? But I don't think that would affect the threshold of the drug release when you apply different ultrasound. It's mainly the nanoparticles itself that would affect in those. Yep, but that's also a good question. Great. I also see another question. Ultrasound energy can affect the integrity of the BBB, and how to prevent this from happening when you try to activate the nanoparticles. Yeah, indeed, that's a very one of the reviewers. I think we indeed get this question from our reviewers during our paper revision as well, and we indeed like try to also did this for the for the for the for the yeah for testing the effect of our ultrasound pressure on the BBB. So in general, because for the BBB opening, people also inject the microbubbles, right? So basically, like if if you don't inject microbubbles, it actually requires much higher, a lot higher ultrasound pressure for opening of the BBB. But so in our case, we don't require microbubbles. So in our supporting information in our in our chemogenetics Nature paper, we actually showing like the basically showing that we don't cause the breaking up of the BBB with our current approach. Yeah, you can see in more details about the conditions of than the experiments of the ultrasound opening of the BBB. Generally, like it requires microbubbles, injection of microbubbles for opening of the BBBs. Great. And I can also see another question. What is the degradation profile of Hopf nanoparticles? And in long-term experiments, will their degradation products cause inflammatory response or cumulative toxicity to the brain issues? Yeah, that's also really good questions. And yeah, I did not manage to talk about it today, but that's also what we have in our supporting information. So basically, we are showing that for the Hopf nanoparticles, like after 5 days, we can still activate the nanoparticles and then to release drugs to do the neural modulations, right? And then the nanoparticles in the brain, they probably will stay there for a couple of weeks. So we did quite a bit of the like toxicity studies in terms of to look at the apoptosis, like a microglia response, astrocyte response. So we look at all these immune biomarkers, and we don't observe much of the cell death or apoptosis or immune response yeah, in the brain, because yeah, we did the local injections in the brain. So we mainly look at the brain, and we did not observe much toxicities. So those are the also in our supporting information in our papers. Great. And I see another question. Will non-invasive regulation cause local temperature increase in the brain tissues? Yeah, and that's also very good questions. And we also did like a measurement of the in our conditions to look at the temperature change yeah, in our activations, and we observed like pretty minimum like change in the temperatures at the relevant like a ultrasound pressure and yeah, I mean the ultrasound we are using in these cases are like a within like the safety region of the ultrasound use. So we did not see much of the like a lot of temperature change over there. Okay, I think we have another question showing that ultrasound might experience energy attenuation when penetrating brain tissues for deeper brain regions, like red brain regions exceeding 9 millimeters, or the larger brain volumes of the humans. How to ensure that the ultrasound energy is sufficient to activate the nanoparticles while avoid thermal damage or mechanical damage of the superficial brain tissues. Yeah, that's also very good questions. So in this case, we use the focused ultrasound. So basically, the ultrasound is we have a focus. We do measure the focus of the ultrasound. So the focus of ultrasound is pretty deep in the brain. So if we do it at 9 millimeters, the focus is around 9 millimeters. So the more superficial, like cortical or like other regions, they actually don't get as high ultrasound intensity. They do get some ultrasound intensity, but certainly not as high as the focus region at the 9 millimeters deep in the brain. So we actually also in our support information we have the pressure profile of the ultrasound, and that's showing that that it's more heavily concentrated at the 9 millimeters, like the deeper in the brain, rather than more like a superficial areas. So that's a but that's a very good point, and and then the way basically the focus ultrasound is the answer. So it's not like a any type of ultrasound would be doing. Okay. And I also see another questions over here. What future perspective do you see to minimize the invasive um, of the nanoparticle applications? Yeah, that's also really good questions. So currently we are doing the direct injection of the nanoparticles in the brain. So it is similar to what we do with like a kind of optogenetics or other like to do the direct injections, right? So it basically it's certainly less invasive than having an implant there for a long period of the time, right? Because that can continue to cause like a immune response and all that. But it does require initially the injections of the nanoparticles that opening a hole in the skull and to inject the particles in the brain, especially for our chemogenetics case. And I believe the future, which we are also working on in my own lab as well. Like basically, I believe that the best way to deliver probably would be the systematic injections, and then to use ultrasound to open the BBB together with the microbubbles, and then that can also locally deliver the nanoparticles into a specific brain areas. And we could people have demonstrated doing that with virus, right? With for delivery of the opsins with ultrasound opening for the BBB strategies, and also some of the other type of nanoparticles that people also demonstrated for the gene delivery. So not quite our nanoparticles yet, but other type of nanoparticles people have done that too. So we believe so that could be a good way to achieving the completely non-invasive. Then it doesn't require any of the opening of the skull or like injections from the skull. So then it would be completely non-invasive. I mean, another alternative could be I certainly know less, but people also talk about like a nasal delivery of the nanoparticles that can also be a potential non-invasive method. But the downside of that, the nanoparticles probably it will be difficult to control where to deliver in these nanoparticles. Okay, yeah. We have another question talking about in the in vivo application on mice and rats. What experiment result demonstrate its potential for clinical translations compared with existing deep brain stimulation, such as DBS, DBS optogenetics and chemogenetics. What is the core competitiveness? Yeah, so basically like a for I guess we have two, right? We have both sono-optogenetics and the sono-chemogenetics. So it's sono-optogenetics. We are our rat experiments, we are able to get in like a cells type specific neuromodulation, and then that really showing like certain long-lasting effect, right? Like which we cannot do with DBS, so basically we compared to DBS, we get cells targeted neuromodulation, and also we do not have an implant, right? Compared to optogenetics as well, compared with both, we don't have like an implant in the brain. We just have nanoparticles, and we apply the ultrasound for these applications. So there's no implant. So and then we are really achieving the treatment of the Parkinson, and have certain like a longer term effects. And with our sono-chemogenetics as well. So and then we are again we are demonstrating in a model like not quite like but in a model like related to depression, for example, or addiction, right? We are showing the therapeutic effect with the for swim model, for example. So that is again we don't have an implant in the brain, not like DBS or optogenetics. We don't have the implants in the brain, and also in comparison to chemogenetics, we are much faster than chemogenetics, right? We only takes a few seconds to for the activation of the selective type of neurons in the brain, and those are like the comparisons. Cool. I think I've probably answered all the questions in the chat, and yeah. Oh, we have one more questions just came up. While the blood constantly flows in the vessel, how to ensure an efficient number of particles in the bloodstream of the region receiving ultrasound activation? In other words, how do you determine the dose of the nanoparticles? I guess so in the motor stimulation, right? And so those ones we did with injection of the nanoparticles through the bloodstream for the neuromodulation, and the later ones we did like a local injection of the nanoparticles. So I think I would assume you are talking about in the in initial case, the motor stimulation. I think basically we believe that I think from our experimental results, the nanoparticles would stay in the bloodstream probably for a few hours, and we generally do it pretty quickly after we inject the nanoparticles, and then the way to see the behavior response is that if we apply the ultrasound, if we do get the motor response, basically we use the behavior response to really to measure the activation, right? So then if I guess if there's no behavioral response, we need to inject more nanoparticles, right? So basically that's what we used for this to answer these questions. Great. Yeah, I think also probably my time is also running up. I probably give back to the RWD. I think I've also answered all the questions, and I will give back to the RWD organizers to continue.
Okay, due to time limitations, this Q&A sections will end here. Thanks to Professor Wang for the illuminating sharing from the groundbreaking development of ultrasound activity nanoparticles to their game-changing applications in the sono-optogenetics and sono-chemogenetics. Every part has deepened our understanding of non-invasive genetically targeted neuromodulation. We are also pleased that our fiber photometry system plays a supporting role in this impactful research, helping to validate the efficacy of the innovative technology. Now more than 8,000 articles have been published using fiber photometry, and it has become a necessary technology for neuroscience research. RWD's fiber photometry system can be applied for a free trial. If you'd like to get more detailed information and try the instrument yourself, we can do a demo. Welcome to contact us. For those who are selected by Professor Wang to answer questions, we will contact you after webinars to confirm the information of the fiber optic news delivery. Once again, thanks Professor Huiliang, even Wang, wonderful. If you still have any questions, welcome to contact Professor Wang by email. We also welcome you to cooperate with Professor Wang and RWD Lab Science. Finally, thanks everyone joining our webinar. Thank you, Professor Wang. Thank you, everyone. Wish you have a nice day. See you next time. Bye.
Bye-bye, everyone. Thanks, everyone. Bye-bye.